gut microbiome experiment, we looked at two gut microbiomes in two different types of dogs, a purebred Yorkie and a mixed breed Yorkie Poodle. In general, a microbiome is types of microorganisms living in a specific location. Specific to our experiment, the gut microbiome is an ecosystem of microbes that help us fight infection, control our weight, and regulate our sleep schedule. According to Coel in his journal, an experiment was done in response to the dietary changes on the microbiome of dogs, humans, mice, and pigs. In his experiment, it showed that diet had a large effect on the gut microbiome. A second study was done by Mascuella in his journal. According to the study, the variable of age on microbiome. The variable of age showed that as age increased, the bacteria present in the microbiome decreased. For this experiment, we posed the question of what is the relationship between the gut microbiome of a purebred dog and between a mixed breed dog. From this question, we developed a hypothesis of is there a difference between the breed type in a purebred or a mixed breed dog that the DNA analyzed will have more microbiome diversity in the mixed breed rather than the purebred dog. A rationale for this hypothesis is based off of animalleak.org. A purebred dog has no other breeds in its family history, therefore having less of a genetic diversity. And a mixed breed dog has different breeds in its genetic makeup, therefore having a more genetic diversity. The first dog we compared was a purebred Yorkie, which was 8 years old, and is represented in our data as sample 795. The second dog we compared was a 3-year-old Yorkie Poodle mix, which is represented in our data as sample 1036. According to our Biology 200 lab manual, our experiment started off with the purification of DNA. The DNA was isolated from the stool samples using the Quiogen kit, and we followed the manufacturer's instructions. During isolation of DNA, a buffer is used to prevent enzymes from degrading the DNA. Unbinding the DNA from the filter is required by an enzyme to weaken the bonds. In order to quantify the DNA, we used a nanospectrometer to quantify and assess the purified DNA to determine its concentration. The PCR was used to produce and measure copies of a targeted sequence. We used a positive control with a known amount of a template purified DNA from E. coli, which is important for the possibility that no other reaction was produced in the product. We used a negative control in our experiment with no template DNA replaced with water. We did this to test for cross-contaminations from other sources. We then used cybergreen as a dye to quantify the double-stranded DNA in the qPCR. The fluorescence is measured at the end of each amplification cycle to determine the amount of DNA that was amplified. We performed 2 applications of the spectrometer. The first application was regular DNA, and the second trial was a dilution. We performed the dilution for the second trial because according to our 260 to 280 ratios, our ratio was less than 1.5, meaning that we had to do a dilution. The results from the first trial of a non-diluted was for sample 795 had a concentration of 6.5 nanograms per microliter, and sample 1036 had a concentration of 6.3 nanograms per microliter. The 260 to 280 ratios for this trial were for sample 795 at 1.73 and sample 1036 at 1.80. The second trial sample, 795, had a concentration of 5.3 nanograms per microliter, and sample 1036 had a concentration of 6.6 nanograms per microliter. The 260 to 280 for samples 795 was 1.99, and for sample 1036, it was 205. From our data, we developed 3 amplification curves from the 3 different primers we used. Our 3 primers used were Eubacteria, Bacteroides, and Firmicutes. Our 3 amplification curves are demonstrated here, and on each amplification curve, the horizontal black line shown here indicates the threshold which the DNA must pass, and all of the cycle numbers are indicated by the x-axis. The x-axis shows how many times the PCR has gone through the amplification process. When the U bacteria was used, which is the first amplification curve, our sample 795 had a 2 negative CQ value of 4.06 times 10 to the negative 6. For the same primer, the sample 1036 had a 2 negative CQ value of 2.45 times 10 to the negative 7. The positive control had a 2 negative CQ value of 2.77 times 10 to the negative 7, and the negative control had a 7.25 times 10 to the negative 9th value. For our Bacteroides amplification curve, which is shown here, our sample 795 had a 2 negative CQ value of 1.45 times 10 to the negative 6. Our sample 1036 had a 2 negative CQ value of 4.77 times 10 to the negative 8. And our positive control had a 2 negative CQ value of 1.49 times 10 to the negative 9th. And our negative control had a 2 minus CQ value of 2.23 times 10 to the negative 10th. Lastly, our CQ values for the Firmicutes, which is demonstrated at this amplification curve, are for our sample 795, our 2 negative CQ value was 1.9 times 10 to the negative 6. For sample 1036, it was 1.33 times 10 to the negative 7. Our positive control was 4.31 times 10 to the negative 9th, and our negative control was 4.74 times 10 to the negative 8. In conclusion, we were successful with our extraction of the DNA from our animal scat. However, it was not successfully quantified. The CQ values in all of our trials were very close to the CQ values of our negative control. With this being said, that means that our amount that we quantified was extremely low. The UV spectrometer did show a presence of DNA, but because of the low results, it indicates that there was a greater presence of RNA in the sample shown by the UV spec. The positive and negative controls were successful for our trials, and from these results, our hypothesis of a higher diversity in the mixed breed was supported. For further experiments, we could use our previous research stating that there is a difference in the microbiome between the ages to test the difference in the microbiome of a puppy or a very young dog to the microbiome of a very old dog to see and show the drastic difference between the two samples. The two animals compared in our experiment were different because of the amount of bacteria found in the microbiome, which is related to age. The age range between the two dogs is 5 years, which supports the research that the microbiome diversity decreases with age. Based off of our hypothesis, our data supports our hypothesis with there being more genetic diversity in the mixed breed dog compared to the purebred dog. For further experiments, we could look further into the microbiome diversity in age, since that might have been a factor in our data. We also could work more efficiently in lab and use larger samples in any further experiments.